Excuse me, Professor.
Yes, please.
Enjoy your class,
Um,
Professor. Sorry for the interruption.
Excuse me, Professor.
Yes, please.
I think actually from the beginning we didn't hear anything.
Okay,
Is
let us
if you
start
don't
from the beginning.
Thank you so much.
Good afternoon, everybody.
Good afternoon, Professor. You can slideshow.
We am sorry about the technical problem. So I welcome you to the last section of this course unit, which is known as parasitology. And at the beginning of the course unit, we said microbiology can be subdivided into subsections, and we also looked at infectious agents of medical importance, which included cellular agents, and these are prions and viruses. We also talked about cellular agents, also known as microorganisms, and these include the protozoa, fungi, helminths, also known as worms, and then arthropods. And then we said usually we prefer to study protozoa, helminths, and arthropods under the subsection referred to as parasitology. So parasitology is actually the study of parasites, and under this study we cover the three infectious agents of medical importance, which I have already mentioned. Let us look at the origin of parasites. It is thought that at the beginning, all organisms were free living, so they could look for their own food. And then they evolved into parasites. And there are evidences show this. For example, when you look at the structure of the roundworm Ascaris lumbricoides, it has close resemblance to the earthworm, which is still free living, while Ascaris lumbricoides is parasitic. So for a free living organism to change into a parasite, it had to go many adaptations, and these included morphological modifications. For example, for those free living organisms that had limbs or structures for motility, found that in the parasitic life, those motility structures were no longer useful. And here we have an example of Plasmodium species, which lack flagella for motility, because as we know, Plasmodium are intracellular parasites, and they readily find their food within the cell. They don't have to move anywhere to look for food. Then among the cestodes or tapeworms, we find that cestodes do not have an alimentary canal, because they are found in the gastrointestinal tract of their host and in already absorbed food by the host, and this food moves to the sestored by osmosis. Then free-living organisms developed structures that were required for parasitic existence. For example, they needed attachment organs. Since the host environment is very hostile, so we find the flukes or trematodes have suckers, while the cestodes or tapeworms have got suckers, and others have got a rostrum for attachment. There were also biochemical modifications. For example, we find that the parasitic species of entamoeba, known as Entamoeba histolytica, elaborates an enzyme known as hyaluronidase, and this enzyme is not found among the commensal species of Entamoeba. And this enzyme is used to penetrate the mucosal surface of the host. So it means those species which are not able to produce this enzyme cannot cause or cannot initiate disease. There were also reproductive adaptations. Since some parasites, some organisms find themselves in environments that are inaccessible. So there was no need to have two sexes or two different organisms with different sexes. So some parasites have become hermaphroditic, whereby both sex organs are found in one individual. And an example is among cestodes and trematodes. Then, to increase their chance of survival, a single developmental stage is able to give rise to many infective stages, which is the case among trematodes and again cestodes. Again, to increase chances of survival, one individual parasite is capable of laying many eggs per day. For example, Ascaris lumbricoides can lay up to 2,000 eggs per day. So even if some of them are deposited in a pit latrine, a few may survive and propagate the species. Then among Filaria worms, there is a phenomenon known as periodicity, whereby the organisms are only found in the blood circulation at certain times of the day, and at other times of the day you will not find these parasites because they hide in internal organs. And the occurrence of these organisms in the blood circulation coincides with the feeding time of their vectors. So this becomes easy for those parasites to be transmitted to the next host by the vector. Another adaptation is to resist the host immune mechanisms. For example, among trypanosomes, which cause sleeping sickness, they tend to change their surface coat so often and this is known as antigenic variation. So each time they change the surface coat, the host recognizes the new variant as a new organism, and it has to start making an immune response against the new variant. And as soon as the immune system is in place for this variant, then the parasite again changes the surface coat, and the cycle continues and continues. In other parasites like the schistosomes, they cover their surface with the host protein so that the host will not recognize the parasite because it is covered in the host tissues. So the host will recognize the surface of the organism as its own tissues, and no immune response is produced against those parasites. And this is known as mimicry. Then some organisms are located inside the host cell. So the humoral immune system or antibodies cannot reach this parasite, so it is protected from the humoral immune response. Now let us look at types of parasites. They can be classified according to location. And here we have ecto, those that are located outside. And then we have endoparasites, these are found inside the host. We have got extracellular parasites, these occur outside cells. The other parasites are found inside the cells, and these are referred to as intracellular parasites. Then according to the degree of dependence on the host, we have got obligatory parasites. If you remove the host, they will die. But we also have facultative organisms or parasites. They will survive if the host is present, but if it is absent, they can still survive. Then we can also classify them according to the period spent on the host. We have got permanent parasites, and the opposite are temporary parasites. They just stay on the host for a short time. Then we have those that are known as hyperparasites. This is where a parasite parasitizes another parasite. Probably here we can only give an example in among the viruses which parasitize bacteria, and these we refer to as bacteriophages, and there is no suitable example in parasitology. Then we have got accidental parasites. For example, we know that the nematode Toxocara canis of dogs parasitizes dogs, but when it produces eggs, and these eggs infect the young dogs, it infects the dogs intrauterine, and when these are produced, the parasites lay eggs, and then these eggs can reach human beings. But because human beings are an unnatural host, the parasite only wanders in tissues, and it ends up being encysted in the host tissues of humans. It does not develop to an adult parasite, so it is referred to as an accidental parasite. Then sometimes we have got what are known as pseudoparasites. This is, for example, when you find eggs of the liver fluke of cows in human feces, although it has been got from the cattle, it is not a true parasite of humans, and it can be easily confused as a parasite. Having looked at the types of parasites, let us also look at the types of hosts. There are those known as final hosts, and these usually harbor the sexually reproductive forms of the parasite. And then they are also known as definitive hosts. We have got intermediate hosts. These harbor the mature stage of the parasite, and within the intermediate host, the parasite undergoes further development. Then we have those known as transport hosts. These ones do not show signs of disease, but when these ones, they are carrying the parasite, and the parasite can only infect the final host when the transport host is eaten or ingested by the final host. Then we have the reservoir hosts. These ones harbor the parasite, and they do not show signs of the disease, but these reservoir hosts are a danger when vectors feed on the reservoir host and at the same time feed on the final host, and then are able to transmit the parasite from the reservoir host to the final host. Here we can give an example of Trypanosoma rhodesiense, which affects both wild animals as well as human beings. Then we can also have accidental hosts. I have already mentioned human beings in case of the ascarid worm, Toxocara canis. These parasites must reach their final hosts through certain mechanisms of transmission, and these include ingestion or feeding. When humans feed on certain foodstuffs contaminated with the parasite. And here we have an example of worms, certain worms. Others use the mechanism of skin penetration. We have an example of trypanosomes, and then we have inoculation, where the parasite through the bite of the vector is injected in the blood circulation of the host. So in skin penetration, probably the best example would be the hookworms. The hookworms, the infective stages of the hookworms penetrate the skin. The other example I had given, trypanosomes, use the mode of inoculation. Then I have already talked about Toxocara canis. In the beach, if a beach is infected, the worms can cross the placenta and infect the fetus, and when the fetus is born, it is born infected and serves as a danger to human beings. Let us look at the effect of parasites on the host. The first one is annoyance, like the mosquitoes. When they are moving around in the room, they cause annoyance. Another effect could be competition for pre-digested food. Among these could be the worms, like the cestodes or tapeworms. Other parasites cause tissue damage. Other parasites can cause allergic reactions, like when worms die within the host, then they can cause allergic reactions. Other parasites suck blood. We have examples of mosquitoes, lice, and bedbugs. Some parasites can obstruct lumens and ducts. For example, Ascaris lumbricoides can block the intestinal lumen, but also the bile duct. And in extreme cases, parasites can cause death. However, a good parasite should not kill its host, because when a host dies, then that parasite will also most likely die. So the relationship between the parasite and the host is not a bed of roses, because parasites have got to meet challenges. They are resisted by the host, and the host can respond. For example, when a mosquito bites a human being, the human being will respond by trying to kill the mosquito with a blow. That's mechanical. Then, where the mosquito has bitten, usually there is swelling, and this is known as inflammation. And the defense cells of the innate immunity move into the area to remove the infecting organism. But we can also have the host developing humoral immunity or mounting antibodies against the parasite. And then we can also have cell-mediated immunity in form of macrophages, natural killer cells, and CD8 cells. Sometimes the host may be overwhelmed and it dies. I don't know if you have a reaction to this first presentation, because there is another presentation. Any reactions to this first presentation? Is there any reaction, reactions, yes, please, Sebunya Isaac. Can I hear from Sebunya Isaac? Yes. There was a question. How do I reach the person asking? The person there is a person asking a question.
The person asking a question. I think that one comes to because sometimes they put it in a chart. A chart. I'm understanding. Yeah, this one.
Yeah.
Yes. So there's this chart where someone they are asking questions. But some of them are just showing muted. So you wanted to look for someone who's asking a question. So they were just sending them information. So these ones are just putting registration.
Want to see. I also wanted to put
Another PowerPoint. Even so these are the messages here.
Yeah, they are muted.
But I feel like evolving from being free, being a parasite is more likely. So I think there is no question here.
Okay, let us move to another.
So you wanted to have another presentation.
Yeah, it is already here.
So we have to stop to share. You close this one. So now Where is another one?
It is already on the,
And then,
or we can get it from, but it was already there.
so we come here.
I guess. Lumen dwelling protozoa. It is here.
It is this one.
Yeah.
I think it is already.
Okay.
Let's check. I think I'm not seeing the whatever, the zoom.
Kale no bazanyisa lot nga
So go to share screen. We said it is
Lumen dwelling. This one.
this one. Yes, come.
Okay. Yeah. Okay, good afternoon once again. Let us go to the last section of this presentation. And it's about lumen dwelling protozoa. The first one of these are known as amoebae, and there is only one species which is parasitic or pathogenic. All the other species are commensal, so they only obtain accommodation, but they do not harm the host. The commensal species include the Entamoeba coli, Entamoeba hartmanni, Entamoeba gingivalis, Iodomoeba bushri, and Endolimax nana. So we shall look at the pathogenic species, Entamoeba histolytica, and it has two developmental forms. The first one, which is the active feeding stage, is known as the trophozoite. And let us look at the structure of the trophozoite of Entamoeba histolytica. It has got two layers.
Recording in progress.
The first one is known as, the outer one is the ectoplasm, and then the inner one is the endoplasm. Amoeba move the trophozoite, actively move by use of pseudopodia, and then they have chromatin. They have one nucleus, and this nucleus has got a small central karyosome. And what differentiates the parasitic or pathogenic amoeba species from the commensal species is that you'll find ingested red blood cells within the trophozoite of the pathogenic species, and these red blood cells are absent among and they are absent in the commensal species. So this is the appearance of Entamoeba histolytica trophozoite. The other stage is known as a cyst, and this is a dormant or resting stage. The mature cyst usually has 4 nuclei, but the immature cyst may have nuclei ranging between 1 to 4. Then there are cylindrical bodies known as chromatoidal bodies, and they are characteristically they have rounded or square ends, and this we find that although Entamoeba coli also has these bodies, these ones look as if you have their edges are irregular, as if you have broken a stick. So this is the appearance of the cyst of Entamoeba histolytica. Let us look at the transmission of Entamoeba histolytica. Transmission is by ingestion or consumption of contaminated water or food containing cysts, and these cysts usually come from infected patients and usually with a chronic infection. But they can also come from individuals who do not show symptoms, and we usually refer these to carriers, especially those who are involved in the business of serving food. However, these cysts may be transmitted mechanically by arthropods, for example, the flies and cockroaches, when they get in contact with the human feces, and then they can also feed on human food. How does the disease develop? The disease is known as amoebiasis or amoebic dysentery, and it can be divided into the acute form, when the cysts reach the intestinal tract. We have said the cyst has got 4 nuclei, and each nucleus develops into an actively feeding stage known as a trophozoite. And then these trophozoites elaborate an enzyme hyaluronidase, which assists them to penetrate the intestinal mucosa. And as they feed, there is damage of blood vessels, and then there is formation of ulcers and bleeding within the lumen of the intestine, and then you have passage of feces containing blood, and this is referred to as amoebic dysentery. Then we also have the chronic form. This is in the form of a granulomatous lesion. There is a lot of inflammation, inflammatory cells, fibroblasts, which develop into a granuloma, and when it is caused by amoeba, it is known as amoeboma. in the rectosigmoid region of the large intestine. This area is the most affected. However, the trophozoites may enter the blood circulation and spread to other organs. And when they enter the liver, they set up a liver abscess, which may pass through the diaphragm and then affect the lungs. And in the lungs, we have development of a lung abscess. The parasite may reach the heart, and here there is development of the heart abscess. It may reach other organs, and wherever it goes, there will be development of an abscess. If it goes to the brain, then you have development of a brain abscess. The clinical signs will depend on the organ system involved. In the diagnosis, we usually collect stool from the affected area, and then we subject this one to microscopy. You can stain with iodine, and then you observe the characteristics of the trophozoites or the cysts, which we already have mentioned. We have said the trophozoite usually has ingested red blood cells. It has one nucleus. The cyst may have from 1 up to 4 cysts, but the mature cyst has got 4 nuclei. Then we can also use serological tests such as immunohemagglutination, immunofluorescence, ELISA, complement fixation, and so on. The control of amoebiasis. The disease can be treated using the drug metronidazole or Flagyl, and we have seen that the parasite is transmitted through drinking contaminated water, so boiling of water or food can control the infection. Water treatment using chlorine can help, and proper disposal of feces in pit latrines or toilets can reduce the rate of infection. We have also mentioned that the common source of infection is food handlers who are symptomless carriers, and these can be checked routinely for presence of cysts. There are some amoeba that are said to be opportunistic. They are said to be opportunistic because they commonly infect or cause disease in immunosuppressed individuals. And one of these amoeba is Naegleria forelli, and the other one is Acanthamoeba castellani. If we start with Naegleria forelli, it has got the amoeboid form, which moves by pseudopodia, but it also has a form that has got flagella. And this Naegleria forelli originates from contaminated water sources. Transmission is by inhalation of cysts in dust. Those are the three forms of Naegleria fowleri. We have the amoeboid form, we have the flagellate form, and then the cyst form. Diagnosis depends on the clinical signs. Usually, the disease is a central nervous system disease characterized by headache, fever, nausea, vomiting, and signs and symptoms of meningitis such as a stiff neck and so on. So the specimens include the cerebrospinal fluid, and when this is tapped, a cloudy, purulent, sometimes containing blood or some purulent fluid is observed, which is usually under pressure, and there is a lot of neutrophilia, or neutrophils are predominant in this fluid. The motile amoeba may also be seen. The other opportunistic amoeba is acanthamoeba. It also causes primary amoebic meningoencephalitis, immunosuppressed individuals. It can also cause ulceration of the cornea, leading to blindness, but it can also cause a chronic granulomatous skin infection. It can also involve the bone, where you have inflammation on the bone or osteomyelitis. Diagnosis is based on detection of the trophozoite and the cyst stage. Control is by treatment using sulfadiazine. We have other lumen-dwelling protozoa, and they are characterized by having flagella, so they are referred to as flagellates. One of them is Giardia lamblia. It has got two morphological forms: the trophozoite stage, which lives in the upper part of the small intestine, but it can also extend up to the gallbladder and the biliary drainage. The trophozoite is pear-shaped with a broad anterior end, and it attenuates towards the posterior. It is bilaterally symmetric, and when it is moving, it displays what has been described as a falling leaf motility. So that is the one on the right is the shape of the trophozoite. You can see the bilateral symmetry, and the one on the left is the cyst. So the cyst is ovoid. It has a cyst wall that is smooth and it is colorless. It has a nuclei So those are the structures that can be found in the cyst. The median bodies, intracytoplasmic flagella, can still be seen within the cyst. Transmission is also by ingestion of contaminated water containing cysts, but also vegetables. The disease is characterized by production of a lot of light-colored fatty stools that are foul smelling. The control is by treatment using those drugs, and then proper disposal of feces, and then boiling of water, and proper cooking of food. The other flagellate is not found in the intestinal tract, but found in the female reproductive tract. It is known as Trichomonas vaginalis, and it causes a disease known as trichomoniasis. This one has only one morphological form known as the trophozoite. It has 4 arterial flagella, an undulating membrane, and 1 nucleus. It has a structure known as an oxyxtile. It moves by wave-like motion. And that is the structure of the trophozoite of Trichomonas vaginalis. Transmission is by sexual contact. Diagnosis depends on clinical signs. In females, the clinical signs include the profuse vaginal discharge, a burning sensation and itching, dysuria or difficulty in urination, which may be frequent, and also inflammation of the urinary bladder. In males, it is usually asymptomatic, although there may be a thin discharge, dysuria, nocturia, enlarged tender prostate, and epididymitis. Diagnosis depends on demonstration of the parasite in a urethral discharge. Can also be serological methods. Control depends on treatment, and it is encouraged that all sexual partners should come for treatment. If it is a polygamous relationship, then all the partners in that relationship should come or should have treatment. The other control method is to abstain if you are not married until you are married, to be faithful, and then use protected sex. Okay, I can see your reactions. Thank you for the supplements. So I don't know if you have any concerns. And this is the end of the presentation. Someone says expected eosinophilia instead of neutrophilia, but eosinophilia are usually encountered in worms.